So I'll just get started on my introduction. I am a molecular biologist by training, and I think, so what that means is I think in terms of molecules of nucleic acids, DNA and RNA mostly, but also my lab works on protein as well. So the overall goal of my talk today is to give you some background on some of these emerging molecular therapies for FSHD that my lab's developing and other people in the field are developing as well. And so that's going to require a little bit of regurgitation of some basic molecular biology that probably a lot of you have had in college or as maybe high school. So I apologize ahead of time for if I bore you with molecules talk, but I think it'll help put you on the same page with what we're trying to do with developing therapies, and hopefully you'll come away at the end of this thinking that it was worthwhile. So just to give you a little bit more background on where we are, this is the Midwestern United States. Michigan is easy to find because it looks like a mitten, but we are located in Ohio here, just beneath Michigan. In particular, my lab is in Columbus, which is almost the dead center of Ohio. This is the capital city. It has about 2 million people in the greater metro area. So I think it's the 14th largest city in the United States. So it's a nice place. Within the confines of Columbus, we have Nationwide Children's Hospital and The Ohio State University. These are linked. My laboratory is located here at Nationwide Children's Hospital, but I have a faculty position at Ohio State. And so this campus is about 5 miles away from my lab. And one of the highlights here is this large horseshoe. It actually is nicknamed the Horseshoe. This is the football stadium for the Ohio State University Buckeyes, very famous college football team in our country. So that's where we live, that's where we work, and what we are trying to do, what we've been doing for the last 10 years or so, is our main goals are to create animal models for FSHD and then use these models to test molecular therapies. And both of these goals are facilitated by, again, molecules, like manipulating molecules, DNA and RNA. And so this is sort of codified here in the central dogma of molecular biology. I think, again, those of you who've studied biology in high school or college recognize this pathway. And the idea is the central dogma tells us about how genetic information flows from our genes and our DNA to make protein, which really makes up the majority of our bodies. So I'll just walk you through this. This is also called the recipe of life. So we have DNA that's in our genetic code. It's in our chromosomes, which is our DNA. And this is present in the nucleus of all of our cells. This DNA codes for a number of different genes. And it's through a process called transcription, it's made into a copy called messenger RNA. And this messenger RNA then serves as a template to make protein. And so we can use what we know about this pathway to manipulate DNA in cells and use those cells to create animal models. And then we can also, and what I'm going to tell you about with some of our work, we can manipulate the sequences of RNAs to target certain genes, in our case, the DUX4 gene that causes FSHD, for degradation. And I'll tell you a little bit more about that in a bit. But the point here is that this whole system here is central to what we do. And so I think just, because this is a fundamental process, I think it's important for us to all get on the same page. So human molecular genetics by the numbers. Here we have a cell. And we have inside that cell a nucleus, and then within that nucleus are chromosomes. We have, in the human body, there are about 37.2 trillion cells. It's an amazing number, hard to think about, hard to conceptualize. Among those, we have about 250 million mature muscle cells, and then maybe another 25 billion or so muscle stem cells. Within each of these cells that have nuclei, we have 23 pair of chromosomes for 46 total. And each one of those contains about 3.2 billion base pairs of DNA. I mean, these numbers are mind-boggling. But this is the sort of genetic code we're talking about here. Within this DNA in our chromosomes encodes about 20,000 different protein coding genes, but then these can be switched around in different ways as well. So we actually have a pretty diverse range of proteins, about almost 100,000 encoded in our DNA. And just a few more fun facts then. So within a single cell out of the 37.2 trillion cells we have, weighs about 6 picograms of DNA by weight. So if we multiply that by the number of cells with nuclei and DNA in them, we're talking about, in US terms, about 11 nickels or 23 pennies of DNA weight in our bodies. And if we uncoil that and stretch every strand of DNA in every cell in our body, end to end, we can go to the moon and back 150,000 times. That's how much DNA is in each of our bodies. And interestingly, if you just took DNA from one cell, so we go to the moon 150,000 times. If you do one cell, you can actually stretch it out longer than the height of Neil Armstrong, who was the first man on the moon. So almost 6 feet long. And this is all packaged within a very small space. A cell nucleus is about 1/10 the width of a human hair. So again, I just want to highlight here that this is what we're talking about with molecules. This really amazing stuff that's in all of us. And what we're trying to do is harness that information and then tweak it so that we can develop therapies for FSHD. OK, so like I said, our understanding of this over the years has produced the ability to identify disease genes, clone those genes, and then create animal models of disease through this manipulation of this DNA. And also then gene therapy and other molecular therapies. And this is, like I said, this is where our lab focuses. And so I'm going to tell you on the next few slides what we're interested in doing and what some of the progress we've made and some of the progress the field has made. So when I started in the field in 2007, this was the state of the field. And I think Natalie Moss already talked about this, how much progress has been made really in a short period of time by our field, partly facilitated by our greater understanding of what's going on in the disease, but also by foundations like FSHD Global and other places that have, like the NIH, for example, that really have started to focus on funding FSHD. So I think we're in this period now where we've sort of slowly ramped up and are now able to have a greater impact, I think, over the next few years. So in 2007, though, the state was we didn't really understand the disease. There were a number of really brilliant people working on understanding how FSHD was caused, but the disease mechanism was still unclear. And as a result, we didn't have a good animal model, or as you might expect, without this knowledge, we can't develop treatments. And I unfortunately don't have the time to tell you all the steps in the molecular mechanism that underlies FSHD because it's such a complicated disease that people have spent decades trying to decipher. But what I can tell you is the punchline, which is Natalie already mentioned, which is that the field is now focused on this gene called DUX4, DUX4, as our primary target. And so what happens in FSHD is that DUX4, this gene, is normally off in muscle. It's not expressed. And then there are triggers, genetic triggers and other triggers that can turn DUX4 on in the muscle. And when it's on, it's toxic, and it can damage muscles, it can kill cells. And so our strategy then is to take this out. We want to eliminate DUX4 expression so it can't have those damaging effects in muscle. And we want to do this through molecular biology. So this is what we're trying to do. But before we can do that, we have to have an animal model to test any therapeutic strategies we want to develop. And over the years, as I mentioned, in 2007, we didn't have a good animal model that expressed DUX4. But over the years now, there have been a number of models developed in all these different species. I'm going to tell you about a mouse model that we're working on that's not yet published, that we think is going to help facilitate therapy development in the field. And this is how we made the mouse. So we took some dirty underwear, and we mixed it with wheat in a jar, and then we waited 21 days, and then we had these mice that expressed DUX4. Is anybody buying that? No. You're looking at me like confused, and I understand because this was a stupid joke. But this is actually a real recipe invented by this guy in 1620 for making mice. In truth, we had to take some different approaches to create our animal model. And I like to put this slide up because this makes it seem so easy to create a mouse model. But in truth, what we had to do was change the DNA of a mouse stem cell and then use that stem cell to generate an animal model. And it was harder than it appeared, than it should have been. It turns out that expressing DUX4 in a mouse cell that will develop into a mouse has to be very tightly controlled because DUX4 is so toxic. If you get even a little bit of leaky expression in that mouse stem cell, you'll never get an animal to develop. So we had to come up with a way to control DUX4 expression so in such a way that we can turn it on whatever we wanted, but make sure that it was completely off as the mice were developing. And unfortunately, we started this project in 2009, and it took us until last year to actually generate these mice. But we have them, and I'll show you what the animals, so this is sort of a big picture image of what the animals look like. Oops. Okay. So these are brothers. They're genetically identical animals, and one has been induced to turn, so we've induced it to turn on DUX4. The other one does not express DUX4, even though it has the capacity to do it. And I think if you just watch them in the cage, you can see the difference between these animals. Obviously, this is the guy we're focusing on here. He's got an unsteady gait. He doesn't walk very well. There's a number of different measurements we can take to measure weakness in these animals, and they show weakness in their leg muscles. They can't rear back. So these are all outcomes that we can measure. And so now when we treat these animals, we can ask whether or not we're improving these. And we think that this is going to be a powerful tool then to test therapies in these animals. And I hope, I hope in the next few years we're able to translate some of what we're doing toward the clinic here. Okay, so let me give you a little bit more background on the molecular biology of FSHD and this DUX4 gene, so we can explain how we're thinking about developing therapies. And just to orient you here, again, we're going back to our DNA here. We have our cell and our chromosomes here. And if we travel down the chromosome all the way to this DNA level, we're talking about a single DUX4 gene down here, okay? Now, this DUX4 gene that we're interested in is located at the very end of chromosome 4 here. So there's all this DNA we talked about earlier in the previous slide. My little fun fact slide. It's amazing that we have all this DNA, and yet it's this one location here at the end of chromosome 4 that we want to key on. So this is the region we're looking at, and if we blow this up, again, here we have the end of chromosome 4, and within that region there are these repeats called D4Z4 repeats that I'm sure many or all of you have heard of or are familiar with. Each one of these D4Z4 repeats contains a DUX4 gene. Now we're focusing in on the very last DUX4 gene. That seems to be the one that's the most important for FSHD. And so we have, again, chromosome 4, we have a series of repeats here. Each has a DUX4 gene, and then now we're looking just at this last repeat here that is highlighted and blown up here in this section. Let me just point out a few key features of what this last repeat looks like. Now we're talking about the DNA level, so this is sort of a schematic diagram of what the DNA looks like at chromosome 4 here when we blow it up at this massive level. So there's this DUX4, what we call the coding gene. This is actually the sequence of the DNA that makes the DUX4 protein. Okay. Upstream of that right here is something called a promoter. This is another sequence of DNA that really serves as the on-off switch for a gene. So if the DUX4 promoter is on, the DNA will be copied into a messenger RNA, and then that messenger RNA can be made into a protein. Okay? If the promoter is off, then the gene is off. It doesn't turn on. I'll come back to the introns parts later, but the bottom line is that here's where the start of the DUX4 gene is, and then the end is encoded in this other sequence that I indicate in this red box called the poly A signal. So every gene requires a poly A signal for it to be stabilized and made into protein. If you don't have the sequence, a gene can't be made. So essentially what happens is you can have a promoter that's turned on. It can transcribe your DNA into a messenger RNA. That messenger RNA has to reach the end of this sequence, and if it has a poly A signal, the messenger RNA will become stabilized, and then it will move to be made into protein. If you don't have this, the messenger RNA gets cut up and degraded, and it doesn't get made into protein. Now, what we want to do then is prevent this sequence from happening so we can't make toxic DUX4 protein. And I just mentioned several steps that have to occur for this to happen. Each one of those then is a node for us to intervene and try to prevent DUX4 from being expressed. And so I'll tell you a little bit about how we're thinking about that. So here's an example where we have this theoretical proteins here binding to a promoter up here at the DUX4 promoter and turning it on, okay? So this, like I said, this will make DUX4 into a messenger RNA, and then that gets made into protein, which is toxic. So, I mean, just it stands to reason that if there are certain proteins that are required to turn this on, then what if we can figure out a way to switch those proteins out and turn it off? So the gene is never, we're just basically taking that on-off switch and turning it to the off position. And in fact, there's a paper published a few years ago by a lab at University of Massachusetts that showed that this can be done in human cells. And so, you know, this is the proof of concept for this in a cell has been shown. And so I think, you know, scaling it up to humans is somewhere in the future. But I think it's a really cool strategy then to, at the DNA level, to try to turn off DUX4 before it ever can be made. So that's one strategy people are pursuing. Another strategy is genome editing. So this is something you may have heard about. CRISPR or CRISPR-Cas. This has been in the news. It's been you can read on the New York Times or lots of other layperson newspapers. So this CRISPR is essentially the hot new thing, and the idea here is that you can manipulate DNA at the DNA level, change the sequence to change, you know, so basically to change any gene you want to in a very precise way. I think that we still have ways to go to make that happen, but the prospects of it are important. And so some people are working on this genome editing approach. There are 3 labs I know of at least now, where what they're trying to do, let's go back to this, we have this poly-A signal that I told you is required for stabilizing the DUX4 transcript. And if you don't have that poly-A signal, the messenger RNA will go away and it'll never be made into protein. So there are labs now that are trying to, at the DNA level, use genome editing to cut this out so it's not present anymore. And then even if DUX4 protein, or sorry, DUX4 transcript is made, it can't be polyadenylated, and therefore it'll be degraded. So this is again, this is still sort of in the future, but it's a path that people are looking at to try to develop treatments for FSHD. In the interest of time, I think I'll skip over this one, and I'll move on to the one that we're interested in doing, that I think is the furthest along in terms of moving through the pipeline to get us to maybe a future clinical trial here. And this is destroying the DUX4 RNA at the RNA level using a procedure called RNA interference, or RNAi. So this is actually a natural process that was discovered by these 2 gentlemen, Andy Fire and Craig Mello. And the discovery of the natural process was awarded the Nobel Prize in 2006. So what is RNA interference? Let's go back to the central dogma of molecular biology here. Again, we have DNA making RNA making protein, right? RNA interference is actually sort of a disruptor to this pathway. The discovery of this, the reason it was so important is because it assigned a new function of RNA to the central dogma. Not only does RNA serve as a template for making protein, but actually some RNAs can also regulate gene expression of other genes. And so this is what RNA interference is. You can have an RNA made from DNA that can disrupt this step here where messenger RNA gets made into protein and actually prevent it from happening. And all cells do this, all eukaryotic cells do this, from going back to single-celled algae all the way up to humans. All of us in this room have thousands of, probably 1,300 or so different small RNAs that can initiate this process naturally in our bodies. And so what we're trying to do is take this natural gene silencing approach that our cells have and turn it on its head, modify it so that we can redirect it to any target gene that we want to. So in this case, our target gene is DUX4. We want to eliminate DUX4. So what we do then is we take these natural RNA interference molecules that are called microRNAs. They look like this. And we can play with the sequence. And this is actually a sequence-specific process. So if we can remove the guts of this thing, of a natural sequence, and then replace it with sequences that target our gene of interest, which is DUX4, then redeliver those back to cells, we can actually retarget these natural RNA interference processes to DUX4 and cause silencing of that gene. And it actually, so we've tested this in mice now, and it works. And I'll show you a slide on that in the next picture. This is just in more detail. Essentially what we do is we have this natural human microRNA, and the places in between these arrowheads then are the sequences that are required for targeting this microRNA to a target gene of interest. And like I said, we just replace these with sequences targeting DUX4. I mean, I'm making it sound easier than it is. We clone these into a viral vector and then deliver them to muscle. And we've done this in our mouse model. And this is an example of what we see here. So if you're not familiar with looking at sections through mouse muscle, this is what it looks like. So this is actually a cross-section through a mouse leg muscle. So if you can imagine a muscle in this direction, we're cutting like this. And so this is just a snapshot of what this muscle looks like. This is what normal mouse muscle looks like, and human muscle as well. You have these polygon shapes. They're all relatively evenly packed, and the nuclei are present at the periphery. We don't see any other intervening cells in there. If we express DUX4 in a muscle like this, we get these inflammatory lesions that you can see here. We have these small cells, and there's abnormal muscle architecture here. Now these are muscles that express DUX4 with no treatment, and then these are the same muscles treated with our RNAi vectors. And you can see we get protection from DUX4-induced damage in these animals. And so we have a number of different outcomes that we've looked at for these. We are at a stage now where we are trying to, so we know that these work, they're functional, and now we're trying to optimize ways to deliver them better and make sure that they're safe. There's only been a couple of human clinical trials using this strategy in other diseases. So it's really an unproven technology. We have to make sure it's safe before we can administer it at high levels to humans, and that's what we're in the process of doing. So here we are, 10 years on, and in 2017 now, the field has made great strides, as Natalie has mentioned. We have a gene target in DUX4. We have animal models with muscle disease that we can use to develop therapies. And I've just touched on a few of the promising treatments in the pipeline. And I think, I'm excited about the one that we're developing with RNA interference. And I think we have a pretty good roadmap for how to move this into the clinic in the next several years. So I'd like to thank again you for your attention, and I'm glad to see nobody fell asleep on me here. And I'd like to thank FSHD Global for their funding support. FSHD Global funded the early stages of this work and some of the early toxicology studies we've been doing that we'll be publishing soon. So we couldn't have initiated this project without them. And then more recently, we've gotten some funding from the NIH to translate this into, hopefully, people in the future. And finally, the MDA gave us a lot of money to help make the mouse model that I showed you here. And that's been an ongoing study as well. So this is where we are. I think the future's bright here in the field, and I hope I can answer any questions that you might have. If we have time for questions, I might have gone a little over. Okay. So, yeah.
You talked also about interference with genes and with RNA and all that. Do you expect any hurdles from an ethical standpoint when it comes to clinical trials and from FDA or TGA instead of bodies like this?
Yeah, so the question is, do we expect ethical issues with ethical hurdles? Gene therapy, so what we're talking about is doing gene therapy. There have been a number of clinical trials already done now, and there have already been gene therapies approved in Europe. There's actually a new one for cancer that's been approved in the United States. So I think some of those ethical hurdles have already been lowered. They were a concern many years ago. Certainly we have, for any trial you're going to do, there are ethical review boards that have to sign off on those. So from that standpoint, there will be hurdles, and I think we'll have to make sure we design a good trial. But from the standpoint of manipulating DNA the way we're doing, I personally don't think that there are any more ethical hurdles to. There's a caveat with that. I mentioned the CRISPR, the genome editing. Again, there's been a lot of hype about that, and I think it's in some ways merited because it's such a cool technology. But what we're talking about is permanently changing somebody's DNA, right? Whereas what we're doing with gene therapy here is different. We're adding a gene back, but it's not changing the chromosome of that person. So there's still a lot of unknowns we have with CRISPR. And one of those is the specificity of it. It's billed as being very specific to this one site, but in actuality, it could probably cut lots of different places. And I highlighted, how many base pairs of DNA we have at the beginning of the talk. So permanently adding something to a cell that can continually cut DNA and change it, that's a hurdle I think that needs to be addressed. So I think from that standpoint, there's still CRISPR gene therapy. I think there's some ethical hurdles, but for the stuff we're doing, not so much, no.
Thank you.
Yes?
Nice to have you here to answer hard questions. How is it that when you delete out all those repeats with DUX4, you've got less DUX4?
Yeah.
And why do we get more activity of DUX4?
Okay, so the question is, in FSHD type 1, which is associated with reduction of D4Z4 repeats, why do we get more DUX4 expression? The short answer to that is that, so to get us all on the same page, on that chromosome 4 region, at the end of chromosome 4, everybody in this room has D4Z4 repeats in various numbers. What's considered to be non-FSHD range of D4Z4 repeats is anywhere from 11 to about 100 on both copies of chromosome 4 at the end there. FSHD, as probably most of you know, is associated with what's called D4Z4 repeat contraction, so that the numbers of copies of D4Z4 repeats shrink from below that 11 threshold. So it's considered, 1 to 10 is considered to cause FSHD. Okay, so if we have this reduced copy number of FSHD, or of D4Z4 repeats, why does that give rise to DUX4 expression? The short answer is that the longer non-FSHD arrays of repeats, those 11 to 100, are embedded in something called heterochromatin. And so heterochromatin is, our DNA takes certain structures, and imagine like a ball of yarn bound up, right? So heterochromatin looks like that balled up yarn. And if you have the chromatin in that state, genes located in that area, which DUX4 is located in the area, can't be expressed. They're not accessible to being made. And so for whatever reason, probably because of the recruitment of certain repressor elements to that region, when you shrink that to this threshold of 1 to 10, that ball of yarn then starts to open up. It starts to uncoil. And when it uncoils, now you have a sort of, imagine this ball of yarn going to like a linear piece of yarn. Now that's open, it's accessible. And now the gene expression machinery that's present in the cells can turn on DUX4 and produce the transcript, which then gets made into protein. Does that answer your question?
So it's de-repressed.
It's de-repressed, yes. Robin, yes.
Thanks, Scott. That's really very interesting. Could I, if you think of the muscle condition in FSH as being perhaps composed of broad, most general terms, muscle destruction on the one hand and a failure of adequate regeneration on the other, I'm just wondering in terms of the timing of when you turn the DUX4 on versus when you give the interfering RNA.
Yeah.
If you cause the pathology and then give the interfering RNA, let's say a few weeks later, will you rescue pathology that's already occurred, or is it simply that if you need to give it at a particular point in time to prevent the pathology coming on? In other words?
Yeah, no, I understand. Right. The short answer is we don't know yet. We don't, so, and the reason we don't know yet is, well, one of the reasons is because we needed this new mouse model to be able to answer that question. So those experiments are ongoing. The question essentially is, can we reverse pre-existing pathology?
Because there does appear to be an impairment of muscle regeneration in FSH compared with, let's say, Duchenne at an equivalent clinical stage.
Well, so I think let me answer this in two parts. Okay, so first of all, we don't know how much DUX4 seems to be a not very abundant protein, even in FSHD. So it's already expressed at low levels. We don't know exactly how much is required to cause muscle damage. Okay, so the question of how much reduction do we need to have to maintain normal muscle function is an open question at this point that needs to be addressed. So can you have some level of DUX4 that turns on in muscle and then you come back in and treat later and lower that before it reaches some toxic threshold? We don't know the answer to that question.
You're affecting the satellite cells, etc., so they're not regenerating the DUX4, etc.
I mean, I guess I'm not certain that we know how much of the satellite cell regeneration actually is participating in this disease mechanism. What I can tell you on the second part, the second part of the answer I was going to give is that with the way that we're doing this strategy, in some ways it is a prevention. So you're right that the way we did this first mouse study, we pre-treated the animals and then turned DUX4 on, right? So we sort of stacked the deck in our favor on that one. If you took a biopsy of a patient, I think you know this, Robin, what we know about DUX4 is it's not expressed at very high levels. And it's been reported in several papers now that if you take a biopsy from a patient muscle with FSHD and then look for DUX4 in the muscle nuclei, you'll find it in about 1 in 1,000 or 1 in every 1,000 or 1 in every 10,000 myonucleus. So if you have this piece of muscle and you're looking for DUX4, it's very rare. We don't find it very often. So the model is that you get bursts of DUX4 expression that are destructive, and then whatever cells that DUX4 is on that then kill that muscle, then those go away, and then the muscle stabilizes, and then you get another burst. And so this burst model is that you have progressive bursts. But DUX4 is not on in every nucleus at any given time. Okay, so from that perspective, if you can think about it like that, if we are able to deliver our gene therapy vector and have it delivered to all these nuclei in a muscle, and it's sort of laying in wait, right? It's there, it's producing its therapeutic, and it's waiting for DUX4 to turn on. In that 1 in 1,000 cases or 1 in 10,000 cases, then maybe what we hope is that it can attack that transcript before it ever gets made into protein, and in effect be a prevention. That's the ideal case, but I think there's reason to think that that's what's going to happen. We have to show it though in our animal model.
Great, thanks.
Yes, sir.
Is there a purpose for DUX4, or is there any underlying reason to have it in the DNA to begin with?
Yeah, there's recent papers that come out just a few months ago this year that suggest DUX4 is turned on in very early development, and it seems to be important for stem cell development at early stages, like maybe 2-cell stage, and then it goes away. What it's doing, I'm not quite clear, but it has a developmental role, and that's when it's supposed to be on, and then it goes off, and it's supposed to stay off in adult muscle. In FSHD, it can pop on in 1 in 1,000 or 1 in 10,000 nuclei, and then it becomes destructive. The only other place that DUX4 has been detected in non-FSHD people is the testes, and what it's doing there is uncertain. Any other? One. Yeah.
Is there any light being spread on the cause of the asymmetry of the condition of FSH on molecular level? Why it might happen? Why this asymmetry?
Yeah, that's a great question. So the question is why do we have asymmetry in FSHD? I don't have a good answer for that. I'm not certain anybody does. Maybe Robin has a good answer for that.
The only comment I would make is that the occasional retinal disease is also highly asymmetrical. But no.
But the asymmetry, to bring up that point, it does, asymmetry and variability in presentation in patients does make it difficult to design a clinical trial, because you want to have predictive outcomes. And we don't quite have those yet, but there are a number, I was talking to somebody earlier, we have a number of people in the United States, clinicians who are developing this consortium of sites all over the country to try to develop usable outcome measures for therapy development. To me, that's a very exciting development, because that wasn't possible just a few years ago. And the fact that it's now an NIH-funded study, multi-center outcome measure development trials, I think is promising. It says that our field's getting ready to take the next step.
Scott, just briefly, do you
Yes.
think that asymmetry could even be starting as early as those few cell divisions very early on in the piece? Could something be going on there?
You could speculate that. I'm not really certain. I don't have a good answer.
Any more questions? Thank you, Scott.
All right, thanks for your attention.